A warm good morning to everyone watching this video. This is Gaurav Kumar Bhargav here. I'm an assistant professor from Department of Pharmaceutical Science, USTM. So today I'm going to speak on immunosuppressants. Now, as the word says, immunosuppressants. Basically, from this term itself, we can understand the fact that this category of compounds, which is immunosuppressants, are the agents which suppresses your immunity. Now, the question lies, why do we even need to suppress our immunity? So the answer to that question is because in many cases, for example, let us take the example of cancer at the very best. During cancer, what happens? Our body tries to fight the immune cells, tries to fight the cancer cells. And as a result, what happens? There is a high possibility of it triggering inflammation and pain all over your body. So, in order to suppress the immunity or in order to suppress all the immune reactions, we use this category of drugs, which is immunosuppressants. Now, in cancer, these are used along with um, of course, the chemotherapeutic drugs. Now, cancer is one example of immunosuppressants. Another very good example is during most of the allergic conditions or autoimmune conditions or any skin conditions, mostly steroids are used. So the steroids are normally used in order to decrease the inflammation or in order to stop the inflammation. The basic function of steroids are to act as immunosuppressants, basically to stop the immune reactions. Now, these are just a few examples of immunosuppressant compounds. So now let us see what immunosuppressants are. So, during an autoimmune disease, the immune system mistakes healthy tissues and cells for foreign invaders. The immune system may essentially turn against the body and attack it. Depending on which part of the body is under attack, the response can lead to different type of autoimmune disorders. Okay, now before I begin with this, I would like to just take a few step backwards to explain about the main terminologies when it comes to immunity. So the first question is, there are two types of cells: antigen presenting cell, which is this part as it is denoted here, APC, which stands for antigen presenting cell, and the second part is the T cell. As we all know, this T cell is a major cell when it comes to immunological reactions. Now coming to this specific cell, APC, antigen presenting cell. Now what is the meaning of antigen? What is the main function of antigen, or which kind of substance can act as an antigen? To answer that question, I'd say that any foreign particles or any particle or any substance which has the ability to trigger our immune system is called as antigen. Any substance or any particles or any foreign objects which can trigger our immunological reaction is called as an antigen. Now, this antigen presenting cell, it can be a dendritic cell or it can be any other type of cells, immune cells. So what is the function of this antigen presenting cell? The main function of this antigen presenting cell is to carry out the information regarding the various pathogens that have entered inside our body and to carry that information back to T cell. So just a kind reminder that all the inflammatory mediators and immune mediators are always getting rise from the T cell. So suppose there's the COVID virus. I'll give the example of COVID virus. So when COVID virus enters inside the body, so COVID virus, if I say about virus, it's nothing but some shells of proteins. So when that virus enters our body, it is the duty of the antigen presenting cell to understand that virus, to understand the structure of that virus, to understand how that virus works, to understand everything about that virus, and to carry that information back to the T cell. So it will react, or it will go and take the information from that pathogen or from that virus, and then it will carry that information back to the T cell. Now the thing is why? So that when the antigen presenting cell carries specific information regarding the specific antigen, the T cell can release specific inflammatory mediators in response to it. So this is just a slight introduction regarding antigen presenting cell. Now today we are going to talk about immunosuppressants. So basically our entire objective of immunity and immunosuppression revolves around this theory. Now, as we can see here, the antigen presenting cell binds with the antigen presenting cell binds with the T cell in two spots, in this complex and in this complex. This is called as the primary binding, and this is called as the secondary binding. So let us see. Let us recall from the first. So as I have said, the antigen presenting cell, the main objective of this antigen presenting cell is to carry out the information from the specific pathogenous cells, or from the specific pathogens to the T cell. And when the APC comes and binds with the T cell, the binding takes place in two spots. The first binding takes place with the CD4 and the CD3 of the T cell. CD4 of the APC and CD3 of the T cell. This is the first binding between the CD4 of the APC and the CD3 of the T cell. The secondary binding takes place between the CD80/86 of the APC and the CD28 of the T cell, and this is called as a secondary binding. Now this is called as the primary binding, and this is called as a secondary binding. Okay. Now after the binding of the antigen presenting cell with the T cell, the first thing that happens is signal transduction. Now, what is the meaning of signal transduction? That means whatever information the APC or the antigen presenting cell has carried, the following information will be transferred to the T cell, and then the T cell will make specific inflammatory mediators in relation to it. So let us see. When the binding takes place between CD4 and the CD3, the signal transduction from the APC to the T cell is done via this MHC, which stands for Major Histocompatibility Complex. Major Histocompatibility Complex. So the signal transduction is taken from this major histocompatibility complex. Now, initially, when this APC binds, the CD4 and the CD3 will bind together, and then the CD28 and CD80/86 will bind, and then immediately the signal transduction will happen through this MHC. Now, when the signal transduction takes place, the first thing that it does is it will increase the concentration of intracellular calcium ions inside the T cell. So again, let me recall, when the signal transduction takes place via this MHC, then the first thing that happens is it increases the intracellular calcium ions. When the intracellular calcium ion increases, then this increase in intracellular calcium ions will cause for the dephosphorylation of Okay, so when there is an increase in intracellular calcium ions, this will activate another compound which is called as calcineurin. So again, I am recalling, when this MHC will transfer, or when this MHC will do the signal transduction, then it will increase the intracellular calcium ions. And this increase in intracellular calcium ions will activate a compound which is called as calcineurin. Now, what is the function of this calcineurin? Let us see. The main function of this calcineurin is to dephosphorylate this N-fat. The main function of this calcineurin is to dephosphorylate this N-fat, which stands for nuclear factor of activated T cell. So, this N-fat is normally in phosphorylated state. So when N-fat is phosphorylated, it cannot enter inside the nucleus of the T cell. But as soon as this N-fat is dephosphorylated, it becomes the dephosphorylated N-fat becomes very easy to enter inside the nucleus. Let us all recall again the steps. At first, the binding takes place between CD4 of the APC and the CD3 of the T cell, which is called as the primary binding. And then there is the secondary binding between sorry CD80/86 of the APC and the CD28 of the T cell. So this is called as secondary binding. Now, after both the binding takes place, then what happens? The signal transduction is carried through this MHC, which stands for major histocompatibility complex, and the signal transduction is carried forward through this MHC to the T cell. Now, the first step this MHC does is it increases the intracellular calcium ions. Now, when it increases the intracellular calcium ions of the T cell, this in turn will activate another compound which is calcineurin. Now, this increase in intracellular calcium ions will activate another compound which is calcineurin. Now, what happens when this calcineurin is activated? When this calcineurin is activated, what it will do? It will dephosphorylate a compound which is called as N-fat, nuclear factor of activated T cell. Now, this N-fat is normally phosphorylated. That means a phosphoryl group is attached along with this N-fat compound. Now, this N-fat is a transcription factor. Now, this phosphorylated N-fat cannot enter inside the nucleus. So, as a result, what we do, with the help of this calcineurin, this N-fat is dephosphorylated. Now, this dephosphorylated N-fat very easily enters inside the nucleus. Okay. So now, inside the nucleus, there are also other transcription factors like activated protein 1, NFKB, nuclear factor of kappa B, then N-fat, nuclear factor of activated T cell, and there are many other transcription factors. So basically, what is the function of these transcription factors? Now, as we all know that inside the nucleus, protein synthesis takes place. Inside the nucleus, protein synthesis takes place. So these transcription factors will tell the nucleus which are the protein it has to synthesize, which are the specific protein it has to synthesize. Okay. Now, when this NFAT enters inside the nucleus, it will undergo transcription and a specific protein is made which stands for interleukin 2, IL-2. It is an inflammatory mediator. Let us all recall again. When there is an increase in intracellular calcium ions, which is a result of signal transduction by the MHC, this increase in intracellular calcium ions will activate a compound which is called as calcineurin. Now this activated calcineurin, what it will do? It will dephosphorylate this NFAT. Now this NFAT is normally in phosphorylated condition, and as a result it cannot enter inside the nucleus. But as soon as this calcineurin removes the phosphoryl group from this NFAT, then immediately the dephosphorylated NFAT enters inside the nucleus. Now this NFAT, and there are also other transcription factors. These are all transcription factors. That means they will tell the nucleus what kind of protein it has to synthesize. So this NFAT will enter inside, and then this NFAT will tell the nucleus to secrete or to synthesize one protein which is called as interleukin 2. Now this interleukin 2 will be synthesized inside the nucleus. But as soon as it gets synthesized inside the nucleus, it will come outside. This IL-2 will come outside. Now, when this IL-2 comes outside, after that immediately it will go out of the T cell. This interleukin 2, first it will come outside of the nucleus of the T cell, and then from this cytoplasm they will go out of the T cell. So when this interleukin 2 they go out of the cell, immediately they will return and then they will bind with the interleukin 2 receptor. The interleukin 2 receptor is located just outside the T cell. So immediately when interleukin 2 goes outside, it will come back and bind with the interleukin 2 receptor. Alright, now after this interleukin 2 goes outside and then binds with the interleukin 2 receptor, what it will do is it will activate one compound which is called as mTOR. This interleukin 2, after it goes outside, again immediately it will bind with a interleukin 2 receptor which is located just outside the T cell. Now when this interleukin 2 bind to the interleukin 2 receptor, they in turn will activate another compound which is called as mTOR. mTOR stands for mammalian target of rapamycin. mTOR stands for mammalian target of rapamycin. Now this mTOR, what it does, it again comes inside the nucleus and then it takes part in the cell division stages. Now with the help of this mTOR, there is purine synthesis. This is a normal phenomena which takes place in cell division. There is purine synthesis and then which leads to the formation of DNA. So this is the normal immunological physiology. Now let us see how the immunosuppressant medication works. So if we see, this is the classification of immunosuppressive agents. The first category is called as calcineurin inhibitors, and examples are cyclosporin and tacrolimus. The second one is mTOR or mammalian target of rapamycin inhibitors, example sirolimus and everolimus. Next one is corticosteroids, example prednisone, prednisolone, and methylprednisolone. Next one, number fourth one is CD80/86 co-stimulation blockers, belatacept and abatacept. So these are the examples. And then the next one, number 5, it comes anti-metabolites or anti-proliferative agents. Example, azathioprine, mycophenolate mofetil, methotrexate, etc. Sixth one is polyclonal antibodies, which is example is anti-thymocyte globulin. Number 7, it comes anti-CD slash anti-CD25 slash IL-2 receptor antagonist. Examples are basiliximab and daclizumab. And the last one, number 8, is miscellaneous non-specific agents. And so let us now see all the mechanism of actions of all the agents. So first, starting from calcineurin inhibitors. Okay, so this category of drug are mainly calcineurin inhibitors, which examples are cyclosporin and tacrolimus. This cyclosporin will come and bind to a protein which is called as cyclophilin. This cyclosporin when we take, it will come and bind to one protein जिसका नाम है cyclophilin. Now this tacrolimus, when we take, it will come and bind to one protein which is called as FK binding protein. Now what happens? Both these proteins, cyclophilin and FK binding protein, in turn will go and inhibit this compound of calcineurin. Now let us look at what happens when this calcineurin is inhibited. Now let us recall the physiology of this part. When the signal transduction through this MHC, major histocompatibility complex, takes place, what happens? It increases the concentration of calcium ions. Now this increase in concentration of calcium ions in return triggers or activates this calcineurin, and this calcineurin will dephosphorylate this NFAT, which then the dephosphorylated NFAT easily enters inside the nucleus. And because of this NFAT, that interleukin 2 is translated and it goes out. So what happens when this calcineurin is inhibited is that this dephosphorylation of NFAT will stop. So what happens when this dephosphorylation of NFAT stops? When this dephosphorylation of NFAT stops, then what happens? There is no transcription of interleukin 2. There is no synthesis of this interleukin 2, and as a result, this interleukin 2 is an inflammatory mediator. It causes inflammation. So therefore, as a result, the inflammation through interleukin 2 will be stopped. This is the main function of calcineurin inhibitors. Now, let us see the next category of drug, that is the co-stimulation blockers, or it is also called as CD80/86 and CD28 co-stimulation blockers. Why is it called as co-stimulation? Because it is the secondary binding. This is the primary binding and this is the secondary binding. The stimulation which takes place via the secondary binding is called as co-stimulation. Now these two drugs, belatacept and abatacept, these two drugs will inhibit the binding between the CD80/86 and the CD28. Now, what happens when this binding is stopped? When this binding is stopped, then the signal transduction is not properly made. So it is very hard to comment on which part is not transducted properly, but we just know from our research that what happens when this CD8086 and CD28 does not bind nicely, then the signal transduction through this MHC, major histocompatibility complex, does not occur properly. The next one is mTOR inhibitors. Now, mTOR inhibitors, what happens, as we all recall, that this mTOR, uh, it takes part in the cell division stages, and it multiplies DNAs. Now, as a result, there is an increase in the number of T cells. So what happens when we stop the mTOR by using mTOR inhibitors? There are two drugs, sirolimus and everolimus. So this sirolimus and everolimus will inhibit this mTOR, and when the mTOR is inhibited, it cannot come inside the nucleus and take part in the cell division stages, and as a result, there is no DNA synthesis, and as a result, the number of T cells, the T cell proliferation does not take place. So after this, the next category is called as anti-metabolites. The examples are azathioprine and mycophenolate mofetil. Now this anti-metabolite drugs, the first one azathioprine, this one is the prodrug of 6-mercaptopurine, which is the main analyte. This 6-mercaptopurine will work on the cell division stages to inhibit DNA synthesis. Now, this 6-mercaptopurine is also used during cancer, so it is also called as an anti-cancer drug. While this mycophenolate mofetil, it will convert into this IMDPH and then inhibit DNA synthesis. So the main target of anti-metabolites are to inhibit DNA synthesis and to stop the proliferation of this T cell. So this is anti-metabolites. The next one is corticosteroids. So there are three examples: prednisone, prednisolone, and methylprednisolone. Now all these corticosteroid drug will come and bind to one receptor which is called as glucocorticoid receptors. Now when they bind with the glucocorticoid receptor, they will go and inhibit these two transcription factor, which is AP1 and NFKB, nuclear factor of kappa B and activated protein 1. Now when they inhibit these two proteins, these two transcription factors are normally the transcription factors which triggers inflammation. So when these two transcription factors are inhibited, the inflammation will automatically reduce, which is the main function of corticosteroids. And then there is one monoclonal antibody, which is basiliximab. Now this monoclonal antibody, when we take, it will come and attach with the IL-2 receptor, interleukin-2 receptor. Now when this interleukin-2 receptor is blocked, at that time this interleukin-2 cannot come and bind with the interleukin-2 receptor. And as a result, what happens? There is no activation of mTOR, and all the cell division and DNA synthesis process will be stopped. And the last one is called as anti-thymocyte globulin. This one is a polyclonal antibody. Now what happens when we take this polyclonal antibody? This polyclonal antibody will attach to the T cell like this in all the part, and then this T cell will be engulfed by other phagocytic cells. Now, as a result, the number of T cells will decrease. So these are the main mechanism of action of immunosuppressive drugs. I hope I have been made clear all the concepts. So thank you. That's it.